Lab 12 senses continued. This is hearing, the auditory system. For hearing and for equilibrium, we are going to look at your ear structure. The ear is made up of the pinna or the auricle, which is the outer part of the ear, whose function is to funnel the sound waves into the external acoustic canal. Now we have a blown up area here of the middle and the inner ear. This is the outer ear. At the end of the outer ear, you will hit the tympanic membrane. The tympanic membrane will then take that air wave pressure and start to vibrate and pass it to the three ear ossicles. The first is the malleus. It's the one that's touching the tympanic membrane. Then the malleus touches the incus. The incus then passes the vibration to the stapes, and underneath the stapes is the oval window. That will then pass that vibration into the inner ear. Your external and middle ear are both air-filled, and one other structure coming from your middle ear, not for hearing but as a pressure release, is your auditory tube, which helps to regulate the pressure inside the middle ear so it is the same as the pressure outside the ear. This pressure will change if you go up in altitude, or you go deep if you dive deep, and the pressure will change, and if we can't regulate it so it's the same on both sides of the membrane here, your tympanic membrane will rupture. Now the sound wave will be sent into the inner ear. The inner ear is going to be made up of your semicircular canals, which are going to be used for angular rotation. We also have some vestibules that are located in this area for linear rotation or linear movement. And then we have the cochlea, this snail-shaped structure, which is where we are going to detect sound. The sound is going to enter at the bottom of the cochlea where the oval window would be located, and then the sound wave will run up to the top of the cochlea on one channel, which is the top tube, and then it will run back down along the bottom tube or canal. These are fluid-filled canals, and in between these two canals is going to be the organ of Corti or spiral organ, which will detect sound. Now looking at this figure, we have a picture of the actual ear model that you would have seen in lab. Again, pinna or auricle, auditory acoustic canal, your tympanic membrane, then you have the three bones, malleus, the incus, and the stapes, M-I-S, to try to remember them in order. They will pass the sound wave into the inner ear. Here's my cochlea and my semicircular canals for angular rotational movement. Looking at the bottom picture, we're just doing an overview on the top of the ear so you can see the cochlea better with the auditory nerve exiting and the three semicircular canals. We can see the tympanic membrane right here and the three little bones that are attached to it. Now with sound, we're going to follow these pictures. We are looking at the cochlea. The cochlea is sort of snail-shaped. It's got a lower bottom layer with the two channels, the up channel and the down channel. Then it's got another layer, then it's got another layer. And the sound wave is going to enter and travel on the up channels all the way to the top and then come back down on the bottom layers, sort of like going in a parking garage in the city. You go up the up ramp and down the down ramp. And in between those two ramps, you have an area, another fluid-filled canal that will house the spiral organ or the organ of Corti. Here we are seeing the two ramps. This is going to be the top, this is the bottom. You go up here, you come back down here. Here is the organ of Corti or spiral organ. This is the spiral organ magnified, and this is going to be made up of two membranes. The bottom is the basilar membrane, and then the top coming out over is the tectoral membrane. Tectoral means to touch, and this is a stiff membrane. Sitting on the basilar membrane, we have cells that are called hair cells, and they have little stereocilia. And when the sound wave comes along and travels along in the bottom channel, it pushes the basilar membrane up, pushes the hair cells up, and the stereocilia will hit the hard tectoral membrane and they will bend, and that will begin the action potential for hearing. This is a figure of the slide that we have in class. Again, basilar membrane is on the bottom. Here are my hair cells with the little nuclei. They're stereocilia. And then coming out, I drew in red so you could see it a little better, the thin tectoral membrane. This is going to be part of the bottom of the upper chamber.